So hey guys, today we're going to be talking about some basic genetic concepts. So to start, we're just going to go through some important terms. So first of all, what even is genetics? It's simply just transfer of genes from one generation to the next. And then a gene is just the part of DNA that codes for a certain trait. And then alleles are alternate versions of these genes. And where these genes are located on the chromosome is called the loci. And we have genotype and phenotype. So genotype, think geno like genetic makeup, so it has to do with alleles. And then phenotype, pheno like physical, so it's just a physical expression of those alleles. I'm sure all of you have heard of Mendel because he's a very important person in the world of genetics. So here are some of his contributions. So first of all, before it was more of a qualitative approach to genetics, but he made genetics move to a more quantitative approach. He also discovered genes are the things that code for traits, and then he came up with two laws. We have the law of segregation, which just states that each gamete only contains one allele. You know, you get your one allele from your mom, and then your one from your dad. And then there's also the law of independent assortment. So alleles for different genes or traits are independent of each other, just because we know with meiosis, those chromosomes independently assort, and so therefore those alleles and genes independently assort. Next, just talking about dominance and genotype. So an allele can either be dominant or recessive, and so dominant is the allele that determines the phenotype, and then the recessive allele is only going to determine your phenotype if it's homozygous. So you have to have two of those alleles for that trait to be physically expressed in an organism. And then we have homozygous, which has one type of allele on the locus. So just think homo, like same, so same type of alleles. And then heterozygous has more than one type of allele on the locus. So think hetero, different, you're going to have different types of alleles on the locus. Now talking about crosses. Crosses simply just means a mating. So one type of cross is true breeding, in which you have two homozygous individuals mate with one another, whether that be homozygous dominant or homozygous recessive. And then you can have a monohybrid cross, which just focuses on one trait, and then dihybrid focuses on two traits. And whenever you have a dihybrid cross, those are going to assort independently because, like we said before, in meiosis, your chromosomes assort independently, so therefore your genes and alleles assort independently. And that just means basically that whatever gene is present, it doesn't affect the other gene. And then for P0, that's just your parental generation. F1 is going to be your filial generation 1, so that's just the offspring of the parental generation. And then F2 is going to be your filial generation 2, which is giving King Tut and colonial royalty energy because that's where the offspring from F1 cross with one another, so incest basically. So that's interesting, but it'd be like that. Now I'm just going to walk you guys through some types of problems. So one example we're going to go through is a monohybrid cross. So again, monohybrid just means you're comparing one trait. So for this, we're going to say there's one trait, which is going to be your seed shape. So round seeds equal dominant, wrinkled seeds equal recessive. And so for this, we're going to true breed for F1 and then cross F1 to find F2. So we know whenever we do a true breed, you're going to have homozygous organisms. So we'll say round seeds, we'll do like a big R, and then for wrinkled seeds, we'll do a little r. So if we're going to do a true breed, again, homozygous, so we'll do this homozygous dominant, and then we'll do a homozygous recessive, and then you just simply cross them. So this is our F1. So all of these are heterozygous because they have the two different alleles. They have the dominant and then the recessive all in one. So now we're going to cross F1 to find F2. So again, they're all heterozygous. So we'll just write that in and then cross. And then, so here we have one homozygous dominant, two heterozygous, and one homozygous recessive. And so I guess if we want to write that out, the one homozygous dominant, the two hetero, and then the one homozygous recessive. So this would be our genotype. And then if we want to do phenotype, again, that's the physical expression. So we either have round or we have wrinkled. So we'll have 3 round, because if you have a single dominant allele, then like we said, the dominant determines the phenotype. So 3 round and 1 wrinkled. So that'll be an example of a monohybrid cross. Okay, so now we're going to move on to a dihybrid cross. So again, dihybrid means you compare 2 traits, and in this case, it'll be dominant is round and yellow, and recessive is going to be wrinkled and green. So we have the 2 traits, the seed shape and the seed color. And then for this example, they want us to cross the homozygous round green with the homozygous wrinkled yellow. Now you can do one big Punnett square. Honestly, that's really confusing to me, so I'm going to show you in what I think is an easier way to do it, with 2 separate Punnett squares, one for each trait, and then you can just use the product rule to find the probability. So we'll use the same letter system for the shape. So round, dominant, wrinkled, recessive, and then green is recessive and yellow is dominant. So we'll do that. So homozygous round green. So round is our dominant, so we have it like that. And the other one is wrinkled, homozygous. So it'll be like that. And then homozygous round green. And then yellow, homozygous. What the heck am I doing? And then so here we have our 2 separate Punnett squares, one for each type. And since we do have the 2 homozygous individuals crossing with one another, that is a true breeding. So I'll just go ahead and do this. So there's that, and then you can use the product rule, which just says you take the probability of getting one trait and multiply it by the probability of getting the other trait to find the ultimate probability. So if we want to find the probability of getting around yellow seed, we would just do round. So all of these would be round. So that's 4 out of 4. And then multiply by the chance to get yellow, which is also 4 out of 4. So that's just the whole probability. And then let's say we want to get round and green. For that, again, 4 out of 4 for that. And then yellow, there's literally no probability of getting yellow. So that's just 0 probability. So that's how you could do the 2 separate and then use the product rule to ultimately find the probability of getting a certain trait. So I did kind of just go over product rule with the previous example, but here I'm just going to go and show you another example and explicitly explain what the product rule is, as well as the addition rule. And these are just shortcuts to kind of make your job easier. So again, the product rule, you just multiply the probabilities of each trait by each other, and you can do this anytime it says the traits are independently assorted, which is typical of your dihybrid, just because, again, like with meiosis in the chromosomes, those independently assort, so therefore your alleles are. So for the first example, Susie's dad is a homozygous short king, while her mom is heterozygous. Her dad also is heterozygous for attached earlobes, while her mom is homozygous for detached earlobes. Find the genetic and phenotypic ratio. And they tell us that short is recessive and attached earlobes are dominant. So if Susie's dad is homozygous short king, that means it'll be the same allele. So he'll be this. And then the dad is heterozygous for attached earlobes. So that's going to be, we'll do these letters for that. And then her mom is homozygous for detached earlobes. And we'll just say she's heterozygous for height because it doesn't say. So then here we'll do our dihybrid cross. So here's that. So then if we want to do the genotype, if we start with homozygous dominant for both, so here I'll write this out. So we know that's going to be 0 because we just don't have any of that. So I guess we'll just skip that. We'll do this one. Homozygous or hetero for both of them. So you have half chance and then a half chance there. So 1/4 probability. And then if we want to say, I don't know, we'll do this one too. Hetero for the height and then recessive, homozygous recessive for the earlobe attachment, so detached. Again, that half. And then also half there, so again, just 1/4. And so that could be for genotype. I'm not going to go through all of them, but also if you want to do phenotype, if you want to say, I don't know, we'll say tall and detached. So we'd have 1/2, and we'll multiply that by, also it just happens to be 1/2 in this example, so like 1/4 of having tall and detached earlobes. So that's how you would use the product rule. Another little shortcut rule that you can use to help you is the addition rule, which this just says if there's multiple ways to get to the same event, you can just add the probabilities. So in this example, it says Timmy's deadbeat dad that left him is heterozygous for dimples. Timmy's mom has dimples. What is the probability that Timmy has dimples that will forever remind him of his absent father? And it tells us that dimples are dominant. So again, his dad that left him is heterozygous. So here's that. And then it just says that his mom has dimples. So his mom could either be heterozygous or homozygous dominant. So that's where we have our 2 ways to get to the same. So we'll do this. Okay, and then, yeah, so it's asking what's the probability that he'll have dimples. So here, there's a 3/4 chance that, oh wait, just kidding. Like he's guaranteed. And then over here, chance to have dimples is 3/4. So then you just add it up and then I'll get your probability of him having dimples. Two other important traits to go over is incomplete dominance and codominance. So incomplete dominance is a blending of phenotypes. So for example, if you have a red flower crossing with the white flower, you'd get a pink flower. In the way I think of it, I just think it's incomplete. So neither of those two phenotypes are expressed. It's more of a blending and in the middle, like an intermediate. And then codominance is when both phenotypes are expressed in an individual. One example is human blood groups. So just think like like both phenotypes are existing together. So there are also sex-linked traits. So heterogametic sex is just having two kinds of sex chromosomes, and that's seen with our males. Just think hetero, different kinds of sex chromosomes because males have the X and Y, and they can also be described as being hemizygous, which just means you only have one copy of an allele in an otherwise diploid organism, just because they have that X and Y again. And then homogametic sex is having one kind of sex chromosome, so this is the opposite, and it'll be seen with the females. So think homo, same sex chromosome, because females have the XX. And then just a little fun fact, turtles and crocodiles' sex is dependent on temperature. So if the eggs are in an environment with a warmer temperature, then it will develop into a male, and if it's a cooler temperature, then it will develop into a female. And then just one example of a sex-linked disease is DMD. It's sex-linked recessive, which just basically means that individual has to be recessive for that trait or disease for them to have it. So if you have any of the dominant alleles, then you will not be affected. Up until this point, we had talked about Mendelian genetics. So now I'm just going to mention fly nomenclature. So this differs from the Mendelian genetics in that traits are named for the mutant phenotype. And so the recessive mutant will be written as such, as like the sin, and then dominant mutant will be written as the psi or whatever. And then the wild type will have a plus with it. And so what the wild type is, it just means it's naturally occurring. And then your alleles are going to be separated with a dash. So, for example, you could do it like this. Again, these are just based off the mutant phenotype. And I think that'll be all for this video. We'll have our quiz on Instagram to test your knowledge and understanding on this topic. And I hope you guys have a nice day.